Hey there everybody and welcome to the Super M Network. Today we are excited to play around with green plants, and when scientists play around, we experiment. The aim of today's investigation is to demonstrate whether these beautiful green plants produce starch. Stay tuned for the rest of the practical. Hey there everyone. So with any experiment, we need equipment. Now the scientific name for equipment is apparatus. For today's experiment, we have a tripod stand. There's 3 legs, hence the name tripod. Sitting below the tripod stand, we have our source of heat, which is the Bunsen burner. Now, on top of the tripod stand, we have the wire gauze. Next piece of apparatus is the glass beaker. This particular beaker has a volume of 600 milliliters. We have a second smaller beaker that is 250 moles. A porcelain plate or a Petri plate, test tube holder to of course hold test tubes, and we have a pair of wire tongs. Now in terms of the chemicals that we'll be using, we have 95% ethanol, which is a type of alcohol, iodine solution, and in my magic flask, regular everyday boiled water. But not forgetting the star of today's show, and that is a fresh green leaf. Now everybody, we are about to start our experiment, but before we do so, the golden rule is to ask for permission and to have an adult present before you conduct any experiment, especially if you plan on doing this at home. Secondly, whenever we are working with a source of heat like a Bunsen burner and we are exposing ourselves to an open flame, we need to take some general precautions. Now, as you can see, my coat sleeves are neatly rolled away because it could easily catch light. Now, if you have beautiful long hair, you will need to know that we can be often distracted and long hair can catch light. So if you have long hair, please do tie it up. Let's begin our experiment. I'll be turning on our gas, or gas, as it is known in South Africa. And I'll also take careful note that whenever I'm working with any type of alcohol, all types of alcohol are highly flammable, especially 95% ethanol. Now, as you can see, I've cleared away most of the apparatus that I'm not going to be using at the moment. I will do so as well and take note of any type of flammable liquids such as ethanol and move them away. Second rule of thumb is that if the ethanol was in this test tube, we would be careful never to expose that ethanol to an open flame because it is highly flammable. And last but not least, when boiling ethanol, it produces a high amount of vapor that we can't usually see with the naked eye. That vapor is highly flammable. Back to our experiment. We have our flame, and something that I just like to point out is that we have a very nice blue flame. The yellow flame is generally hot, but the hottest part of the flame is the beautiful blue that you see below. We have a tripod stand and we have our wire course. Now we will take our 600ml beaker and my magic flask. Because nobody really enjoys watching water boil, which is the same as watching paint dry, I've taken the liberty just to prepare some boiled water. We'll be filling up this beaker with approximately 350 ml of water. Now we will take our beaker and place it on top of our wire gauze. And you guessed it, we're going to wait for this water to boil. Now that our water is boiled, you guessed it, it's time to take our beautiful fresh green leaf. Now you've noticed that I am wearing nitrile gloves. That is just to protect any type of starch that might be present on my hands, contaminating this particular leaf. So we will take our fresh green leaf and we will place it into our boiling water for approximately a minute. Now, whilst we are waiting for our minutes and I set my timer, let us consider why is it that we need to place our green leaf in boiling water. I wonder. Now that our leaf has boiled in boiling water for approximately a minute, I will turn off our heat source. Now you will notice that the color of the water has changed. The color of the water at this stage does not have any bearing on our experiment. It just so happens that this particular species of leaf was obtained from a very high and special mountain which produces a very special type of plant. So we've now removed our leaf from the boiling water and we will proceed to the next step of our experiment, remembering to take extra caution when working around 95% ethanol. Now for the next step of our experiment, I will take a test tube Now, for the next step of our experiment, we will take our freshly boiled green leaf and place it into a test tube. like so. We will then, away from the heat source, even though I know that we are taking a precaution and it is off, cover the leaf in this test tube with 95% ethanol. We will now take the test tube with the freshly boiled leaf covered in ethanol and place it into the boiling water or previously boiled water, and we will notice the following. The ethanol will slowly begin to boil. Why is the freshly boiled leaf now boiled in ethanol, I wonder?
So now we remove the test tube with the ethanol and very carefully we try out our decolorized leaf, also remembering to point the test tube away from yourself or any other learner that could be in the way. Here we see our decolorized leaf. Just take note of how stiff and actually brittle that leaf is. It's because all of the chlorophyll as well as the moisture has now been removed. Next step of our experiment is to hydrate the leaf in a bit of warm water. We can now remove away our heat source, just preparing our Petri plate with our iodine solution. For the most important step probably, and that is the starch test. I'm just spreading out that leaf. You can have a look that most of the chlorophyll has now been removed from that leaf. Now I've taken a couple of drops of the iodine solution and I've actually placed it onto the leaf. See in the background. Apologies for not getting that in the video clip. And as you can see, a beautiful positive test for starch indicated by a blue-black color.